Welcome to the DNA Advantage podcast, where you can learn facts about your DNA that can help you live a healthier, longer life, doing all the activities you dream of doing. If you want to gain more control over your health and the factors that influence it, you're in the right place. Hi, this is Lisa Witherow. Today we'll be discussing MTHFR, methylation, and their impact on our health. I've received lots of questions about MTHFR, and there seems to be a lot of confusion about the importance of MTHFR and methylation. This podcast is intended to give a little bit more explanation, so it will be a little bit more technical, but it should offer a clearer view of how the system works and why multiple genes are involved. Methylation is super important because it is necessary to make neurotransmitters, metabolize neurotransmitters, make cells, which is a critical process for fertility, growth, and healing. Methylation is important for making our DNA, regulating our gene expression, making hormones, clearing histamine, detoxifying heavy metals and other toxins, making energy, helping our muscles contract, making creatine, and the myelination of our nerves. MTHFR is one of the genes that is discussed and emphasized when it comes to certain health conditions. And it's important to recognize that MTHFR is not the only gene that should be analyzed and understood. There are two single nucleotide polymorphisms, or SNPs, under the MTHFR gene that have been researched extensively and are emphasized that are critical to the methylation process. MTHFR A1298C is required for the production of tetrahydrobiopterin, which is BH4. And MTHFR C677T lowers homocysteine using B12 or methylcobalamin. Some of the other genes are CBS, which removes homocysteine from the methylation pathway, COMT or COMT, which metabolizes dopamine, norepinephrine, epinephrine, and estrogens. FUT2 or FUT2 maintains diversity of intestinal microbiome and is protective of B12 status. MTR methylates homocysteine to methionine. MTRR reactivates B12 and helps create SAMe. MAT, or MAT, converts methionine to SAMe. TCN2 transports B12 from plasma into the cells. And BHMT is a shortcut for homocysteine to convert to methionine. There's a lot to learn for each one of these genes, so I'm really just glossing over them, but I give an overview and explain the function of each of these genes because it does give a better sense of why we can't just look at one gene. It really is important to understand how one serves the next, and in some cases, they work together as a team. So it is something to keep in mind that you cannot just isolate a gene and say, all of my problems are due to this one gene, or I'm just going to take this one supplement and that's going to solve the whole problem. Because as much as I would love to be able to be prescriptive with this and say, anytime you have this gene, then you take this supplement and everything is rectified. Unfortunately, that is not likely, and this is not a diagnostic or prescriptive process. So when you're looking at the genes, it's super helpful. It isn't something that you should take lightly and say, this is too complicated and it isn't something that I can ever figure out. It definitely can be figured out, but each person is different. The genes are working together, and there are important facts to know about each person, and then those puzzle pieces come together and you start to see the bigger picture and how your system can be supported and symptoms can be eliminated and disease can be avoided. So methylation is the transfer of a methyl from one substance to another. Or as an example, it transfers a methyl from a nutrient to your DNA. That allows the DNA to express. So that is something that if we have a breakdown in methylation, because methyl donors are either not there or they're not being transferred appropriately, there's a breakdown in the process and our bodies don't function appropriately or optimally. The main methyl donor is SAMe, which requires the methylation cycle to be fully supported by nutrients like active folate, B12, B6, B2, magnesium, betaine or trimethylglycine, and vitamin D. When there isn't enough SAMe, the production of glutathione, coenzyme Q10, serotonin, melatonin, nitric oxide, norepinephrine, epinephrine, L-carnitine, cysteine, and taurine can be impaired. We can experience the condition of undermethylation and overmethylation. Depending on our environment and our nutrition and our lifestyle, those are things that can influence our methylation aside from our DNA. We need to be aware of the condition or the status of our DNA in order to understand what nutrients, lifestyle, and environment are best for us and the support that we need. According to Dr. William Walsh, 22% of us experience undermethylation. It can be caused by SNPs in the methylation cycle, if we have histamine overload, or if we have low methionine, which then leads to low SAMe, and this is often caused by low protein. Overmethylation is less common, but about 8% of the population has a tendency for this condition. Overmethylation can be caused by impaired creatine synthesis and SNPs that lead to reduced creatine. Because undermethylation is more common, we'll focus on that today. There are 2 MTHFR SNPs that we've already talked about that have been studied extensively. And when we talk about C677T, it's responsible for the folate metabolism. If it isn't functioning optimally, it can compromise the use of folate and other B vitamins. It can interfere with proper homocysteine recycling, limit the detoxification process, and may impair proper protein synthesis, leading to various nutritional imbalances, including vitamins, minerals, amino acids, and fatty acids. The status of a heterozygous C677T for MTHFR is about 30 to 40% loss of function, and homozygous for C677T MTHFR is about 60 to 70% loss. But you always want to look to your health status, and you always want to look to your symptoms and see if you may possibly already be compensating enough and supporting the body through nutrition and environment and lifestyle to support this. So you don't automatically start supplementing because you could then overmethylate. Symptoms of MTHFR C677T can include high homocysteine, cardiovascular disease, stroke, heart attack, deep vein thrombosis. If you have a family history or you have personally a health history or current status of these symptoms, that's what really confirms that something needs to be done to support your system. If you have a mutation in MTHFR A129-8C, if it's expressing, it's more commonly linked to neurotransmitter disturbances and nutritional imbalances, especially compromising amino acids. the more common symptoms for this SNP are depression, anxiety, panic disorders, ADD, ADHD, learning issues, and behavioral disorders. A heterozygous status for MTHFR A1298C is about 20 to 30% loss of function, and homozygous is about 40% compromised. So again, it's not assumed that because you are heterozygous or homozygous that you're presenting with these symptoms. But if you are presenting with these symptoms, then this is a direction to be looking and analyzing, and even looking at some of the other SNPs that we'll be talking about to understand what is going to be best to support your body and avoid disease, avoid continued symptoms, and the malfunction of the enzymes in the future. The best first step beyond a whole foods diet is to support your body with a high quality multivitamin. And then the second is to have a good B supplement. A good B supplement is not going to include cyanocobalamin because cyanocobalamin is a synthetic version of B12, and it does have a cyanide molecule to it. So when the cobalamin is utilized by the body, it leaves behind a cyanide element, which can cause issues with health. You can get natural sources from meat, red meat especially, but it is something to take note that there are multiple types of B12. So you have, of course, cyanocobalamin, which I just mentioned, and then there is methylcobalamin, hydrocobalamin, and adenosylcobalamin. In order to determine which of these is best for you, it's important to look at multiple genes. COMT is one of those genes to recognize your status, as well as a gene that we haven't mentioned in this podcast, which is VDR, the vitamin D receptor gene. There are some considerations when deciding which version to be taking and which is best for you. Of course, it's also very good to eliminate sources of the synthetic version of folate called folic acid because it can block receptors. Not everyone is impacted negatively by folic acid, but it does require additional work for the body to convert folic acid to the usable active version. So it is advisable to avoid folic acid when possible, especially if you have an MTHFR SNP. The other considerations for undermethylation that I mentioned involve different SNPs we can discuss in detail in another podcast. But histamine elevation can be caused by SNPs, environmental factors, and diet. So we need to be aware if we have symptoms of histamine elevation, if we have a runny nose and allergic reaction to things, if we have even food sensitivities, we should be looking in that direction of histamine intolerance and the DNA SNPs that are involved. Also, we need to look at the methionine cycle and understand how that contributes to our health. And we want to make sure that the other SNPs, which are MAT, CBS, MTR, MTRR, and BHMT. If we see a theme and we understand our DNA has multiple compromises in those DNA SNPs, we need to be aware of that and start looking at ways that we can support SAMe. And it isn't necessarily to take SAMe. We have to look at which of the SNPs and the conversion pathways. Do we need methionine? Do we need SAMe? Do we need cofactors like B6 for CBS? Or do we need B12 for the MTR-MTRR process? Also, the BHMT is supported by betaine or TMG, trimethylglycine. So that is another area that we can support. When there are multiple SNPs in different systems, it's important to recognize if there is a backup in one particular area before you start taking methyls. As an example, when you take methyl donors, the byproduct of methyl donors include sulfur. And if the sulfation pathway is not appropriately functioning, there needs to be consideration to improve that pathway before you start increasing methyls, or you can have an adverse reaction, including things like anxiety, irritability, sleeplessness, tired and wired type response. But it is important to recognize that this is not because you should never take methyl donors. And the same is true for sulfur. People who eat sulfur vegetables or take certain sulfur containing substances will respond and believe that they can never take or eat sulfur vegetables. And sulfur is a component that's very important for the body. So you don't want to jump to that conclusion and neglect to supply that to your body. You just need to recognize what is blocking the metabolic process of sulfur to make sure that the proper processing is taking place. This is just a brief overview of methylation and how it contributes to health and why MTHFR is targeted as the gene that could make us sick. But it is so important to recognize that we can't put all the blame on one DNA SNP. We can't look at one gene and assume that that is the only thing we need to concentrate on in order to feel better. It is more complicated than that, which is not necessarily good news. But the good news is that if you are struggling with health or with certain symptoms or even concerns of disease, there are multiple areas that you can look to gain knowledge about your own body and gain control over the factors that you're most concerned about so that you can improve the condition and make sure that you have the support that your body needs for health in the future. If you enjoyed this podcast, please subscribe so you can get notification for all the new episodes. And if you have questions, please post them on the DNA Advantage Facebook page. Thank you so much, and take good care of your DNA.